So, hi everyone, and welcome to today's discovery session. These sessions are meant to support students, businesses, and other organizations with an understanding on how to use data and artificial intelligence. Before we get started, I would like to begin by acknowledging that DeepSense works out of Mi'kmaq, the ancestral and unceded territory of the Mi'kmaq people. This territory is covered by the treaties of peace and friendship. And we would like to acknowledge the Indigenous people whose traditional lands we are on today. And we also want to ask everyone takes a moment to consider the territory in which they are situated and their own personal responsibility in the processes of reconciliation. My name is Brogan, and I'm the communication specialist with DeepSense. So for those of you who don't know, DeepSense is a project based out of Dalhousie University in Halifax, Nova Scotia. And while our home is Dalhousie, we actively partner with universities and colleges across Atlantic Canada, and our focus is on working with companies in the ocean sector who want to leverage data to create a smarter and more sustainable ocean economy. We connect these companies with the next generation of AI, machine learning, and data experts, which is our students. And together we design projects tailored to the specific needs of the company and then match them with a student that will execute the project. These opportunities can take the form of master's research projects, co-ops, or internships. Past projects have covered a wide range of topics, including data and analytics, collection, visualization, cleaning, processing, as well as AI and machine learning. So we've done all kinds of things ranging from fish tracking to spotting whale shapes from aerial images that can be used to then possibly lead to temporary fishery closures when there are right whales or other endangered species in the area. So if you are looking to expand your skills, work on real-world projects, and gain valuable industry experience, be sure to submit your information to our student pool. Projects, internships, and co-ops come up throughout the year, so it's a great idea to get your name in early, since this is the first place that we look when we're filling positions. And I'll share the link for this in the chat. And also make sure that your resume is top-notch, because only the candidates selected for interviews will be contacted, so you want to make sure that you stand out. Now, for today's session, shellfish growers are in need of long-term verifiable biological, environmental, and production records. IntegraSEE and DeepSense have developed AI models to recognize open and closed mussels and oysters and generate quantitative red, green, blue values from video images. The goal is to create a novel biosensor to better understand shellfish production from the standpoint of the product and its environment. Today's session is led by Lawrence Taylor and Najwan Hassan. Lawrence Taylor is a biologist with a Master of Science, an entrepreneur, and CEO of IntegraSEE. He began his career as a freelance biological diver and underwater photographer for marine research and television productions. In 2016, Lawrence shifted to ocean tech, realizing the untapped potential of vision-based oyster graders for generating both production and biological data. His innovative approach has earned him recognition, including wins at the Louisbourg Seafood C++, Halifax's Fishhackathon, and participation in accelerators such as Propel ICT, Dal LaunchPad, and Blue BioValue in Portugal, where he showcased IntegraSEE's novel biosensor technology. Najwan Hassan is a computer science graduate who recently completed an IT programming for artificial intelligence degree from NSCC. She brings 3 years of experience in introducing products and services to emerging markets. With a robust foundation in machine learning and a passion for artificial intelligence, Najwan is well equipped to deliver transformative AI solutions that enhance strategic business initiatives and secure a competitive advantage for clients in the technology industry. If you have any questions throughout the presentation, we'll be taking them at the end, but feel free to drop them in the chat at any point during the presentation. Now, that's enough from me, so let's hear from Lawrence and Najwan.
Great, thanks so much, Brogan, and hello, Najwan.
Hello.
What I'll do is I'll start off with a little brief background on my life and how I got into all of this. What has been interesting, what I found at a couple of hackathons was that there are all these brilliant students like Najwan who know the computer science element, but don't particularly have the background in the field of fisheries and aquaculture and the marine environment. So that's what I've generally worked on my entire life. I grew up in a family where my father was a freelance cinematographer and my mother was a journalist, so I learned to tell stories at an early age. Completing an undergraduate degree at the University of Guelph, I took a broad spectrum of biology courses, so I could cover everything from bacteria up to elephants and tigers. But I also, my first year at Guelph, immediately my roommate and I took a scuba course. So I've been scuba diving since 1979, I hate to admit. So when I moved to Halifax, I took my master's degree where I was looking at a fish parasite model where I had to spend many hours collecting snails with my parasite in it and then infecting fish. But when I graduated that, I was really pleased because I stepped into a 3-year sea scallop project. It was part of OPEN, the Ocean Production Enhancement Network in the early 90s. So our job was to take very small scallop seed, distribute 10,000 of them across a research site. And then my job was then, as you can see in the top left, take my camera, go down to the bottom and do video transects. So I soon learned what it was like to be an ROV. But one of the biggest lessons I learned during that was 3 hours of filming underwater took me about 5 days, 8 hours a day to then go through all the footage manually and then count animals on the bottom. The other thing I learned too is that in the bottom left, you'll see that's what the camera would see. I put a little wand, as I called it, with black hash marks on it, so I had a length measurement. But because of the camera view that I had, as I passed over the bottom and a large scallop showed up, I actually couldn't tell dead ones from live ones, the dead ones we call clunkers. So I soon realized there's lots of information that video can produce, but if it's not answering the questions you need, then you need to address the camera setup that you're using and the rest of the methodology. So I realized that in order for me to complete this, because we couldn't change this, I had to work with one camera, I actually went through and would point out all the dead ones as I was diving on the bottom. So that was important for me. The other part that sort of follows along with this, and jumping forward a little bit, I worked in a post-production house helping manage programs through the system. And I soon learned that for producers and directors and so on, what they really needed to know was, and for researchers, what's the question, what's the story you want to tell, what are the formats you want to deliver in, and the deliverables. And then you sort of reverse engineer back through. So again, if I knew that I wanted to know number of live and dead scallops, I really needed to retool as going back through and have a 2-camera system, one low, one high, and that would have accommodated for. Pushing through in my career, so after the Open project, I then worked as a freelance underwater photographer for science and TV. The top right is I actually, because I had worked with fish so much, I created a wooden female salmon dummy. And what I wanted to do was take it onto the spawning ground, set up cameras, and then animate the dummy to see if I could actually get male salmon to spawn to the dummy. And sure enough, I did. It says something about males in spawning time. Anyways, so I've used a couple of different setups, worked in freshwater marine environments, and one on the bottom right is Wilma the beluga whale, one of my favorite shoots, which I did for a number of TV shows. And again, animal behavior was really what I was looking at and trying to record. So that's a bit of my background. But in 2015, if I can move ahead here, I realized after working in the post-production industry that I wasn't getting much work as an underwater photographer. So I was looking back at science and I came across on the top left there, you'll see that's a oyster grading machine and it uses vision technology. On the right-hand side is a belt. Oysters are separated into a single line. They pass through that big box in the middle. Video system then looks at them and essentially does measures length, width, and height, and then analyzes what sort of grade it is. So when it pops out on the left-hand side, you'll see 4 little distributors there. They pop into their correct grade. Well, I realized it's like having little scientists in a box who can analyze an oyster in a second. So it was giving length measures, width, and height. What were they doing with that biological data? So not just for food processing, but on the biological side, they could infer that data back into the catch or that harvest. And from that harvest, they could then see maybe if it's a very large production site, if there were changes or differences in growth within their own lease site. The other two is that if you had a production year where you're growing animals in 4 years, the animals you harvest had similar growing conditions as the next group who would be harvested the following year for 3 years. So time over time, you could build up quite a strong bit of information as to the biology of these animals in your grow out site, let alone measuring them on the site in real time. So that was always in the back of my mind. I then went off and participated in the Louisbourg Seafood C++ competition. And there where I was really pitching, you can do seafood processing and biological data gathering at the same time. They don't have to be two different events. At about the same time, I came across a book by Thomas Freeman. I have it beside me all the time and I refer to it for quick reads. The book was Thank You for Being Late. And what you'll see there in the middle are cow's legs. And the story he gave was by simply putting pedometers on cows' legs, the amount of information computer scientists got about the cows was phenomenal, even though they knew nothing about cows. So what happens is, when cows start to walk faster, they are usually approaching estrus, or at least they found as estrus came on, the cows started to walk faster. From the data, they further learned that if you coordinated it in the first 2 hours, you were going to get 75% chance better of getting a milking cow. The next 2 hours, you get a bull. So obviously they want cows versus bull in the milking industry. So I thought that's the great thing about the technology that's coming through. Could I exploit it? So there, over on the right, on the top, I worked with Professor Alex Martinenko in Truro through Dalhousie, and we took a 2-camera system. We were starting to analyze oyster grades, so we could look at height, width, and depth of the animals. That didn't go too far. I was a little ahead of my time there. Just below that, on the bottom right, I worked with Professor May Sato, and at that time, DFO, Department of Fisheries and Oceans, was asking lobster fishermen to now take bycatch notes, which meant that they had to have an observer on board, which is often fairly costly for the lobster growers. So I thought, let's try and make a little slide. So if any undersized animal or bycatch would be placed at the top of the slide, then the bottom of the slide was over the edge of the boat. As the animals went down past the camera, it would take a picture. We went through and recorded about 300 lobsters so that when they passed through our system, we could actually do basic length measures of the lobster and also determine male and females. We just needed to follow up with fish. We were a little ahead of our time again with that. So those 2 projects, at least I knew I was going in the right direction. And I thought, well, where can we go next? So, excuse me, marine mussels have actually been used as sentinels, indicators of sort of water quality and what's going on in the marine environment for quite a long time. NOAA started what was called a Mussel Watch program that 60 countries across the world participated in. The only problem is that they took live animals and then had to do tissue sampling in order to get an idea of what the animals were collecting, whether it was, most of it was sort of pollution elements that we were looking for, but you can also look for viruses and that sort of thing. So that's a destructive sampling method that's kind of hard on the mussel. What I looked at and was introduced to that we could do this without actually destroying the animals, and the animal's behavior actually could give us an indication of water quality. So I was a little leery about this at first. So then I came across this. So on the top right there is a picture of mussels in a wastewater treatment plant in Poland. And so 8 million people and the plant managers rely on mussels to indicate that there's something going on within the water, and that you can then go through your barrage of water quality testing to figure out exactly what that is. So they're actually being used within wastewater treatment plants and a couple other examples within North America as well. Now, the thing is, DFO has used these in the wild to look at the reaction of mussels to hazardous algal blooms. But again, like the wastewater treatment plant, the 2 bottom photos, well, especially the bottom right, is a Hall sensor is usually attached to the animal. So that's a magnet on one shelf, the Hall sensor on the other, The sensor then can determine how much the shell is opening by a single degree based on the magnetism that it's detecting. Now, talking to the DFO researchers, I said, well, what if we do this with video? And he said, well, that's great, because then you get a visual context of what's going on in the marine environment. At the same time, you're looking at the animal's behavior or shell movement. So that's where then we moved into our first prototype. So in 2021, as a cameraman, I always love a challenge. I have to go film animals underwater. So all the elements were essentially off the shelf. I took a Blue Robotics underwater enclosure, took a Raspberry Pi, put a couple of fans on it, and then put a WEC connector out so that we could put their system in front of mussels, which you can see in sort of the top right of that picture. And the camera is then monitoring the animals in real time and recording the video. What was fun about this system is that I could actually sit at home because the Raspberry Pi camera system could only collect 18 hours of footage. I could sit at home every morning. I would dump the video files and then reset the camera and start the recording over again. I could also then take a look at the last day's footage, which was interesting because the very first day we put this system onto the Stella Maris platform, which some of you may know, went into the water in 2021 for about a year. I bothered the chaps who were running the system and said, all right, so 9 of our mussels all closed down at the same time means something was going on underwater at that time. What does your data say? And of course, they said, don't bother us. We haven't collected all the data and haven't got all the sensors together. So this is the great power of mussels, is that they live in this environment. They're very sensitive to what goes on within the environment, and they'll react to it very quickly. And they also have a little bit of a language. So that's why we wanted to move into DeepSense to then determine, can we actually get vision AI, machine learning, to recognize an open animal and a closed animal, so that when mussels are exposed to hot water or cold or lack of oxygen, they will close right up and stay closed for a while. Whereas when they are subjected to a hazardous algal bloom, they do something what's called a micro closure. So ours was then to look at, can we recognize open and closed animals and start our journey on understanding what they're saying. At the same time that year, I happened to teach underwater photography at NSCC's Ocean Tech course for 1 year. And so I came back and asked students, I gave them on the top right there, you'll see one of my old underwater housings. So they stuck a Raspberry Pi system in, and we just wanted to see if infrared lights could be used to record at night. So our system on the left could only record during the day unless we put floodlights on. And so the system on the right, they were testing to see if infrared lights could be used at night for recording to replace artificial lights, and it did. So those are the earliest prototypes and testing. Amos then, and I hope I said her last name correctly, she then worked on for the next year. She took a year's worth of video data, worked on developing the models then, and could detect open and closed animals. What was interesting, because there were so many open animals most of the time, the data was a little bit skewed on the closed ones. So that was really the only adjustment. But if you want to see more about Amos's work and how she started it, there is a DeepSense session called the Marine Canary Ocean Health Monitoring Using Mussels and Computer Vision that you can go take a look at to see what challenges she had during her participation with us. So next one. We have their system then, which can be placed in the ocean, recording mussel behavior. But we started working last year with Clean Valley as part of an ocean supercluster project. Clean Valley has a setup in Portugal. So on the picture on the left, that big building then contains fish tanks. There's a startup there that's working on developing the process for growing corvia, which is a common fish eaten in Portugal. So the wastewater coming out of that plant then goes into Clean Valley's little trailer there. So the center shot is one of these trays where this wastewater comes in. Clean Valley is then growing specific algae species to then, on the picture on the right, to feed oysters. That's a special grow system for growing the oysters. So to better understand algae in the presence, because hazardous algal blooms are a big issue, I thought, why not take our camera system, our field system, and split it so that we could analyze within the video we were recording color in relation to algae, which is related to chlorophyll, and then animal behavior separately. So that's where Najwan really came into play, because I went, all right, we're going to change species now. We're going to move from mussels to oysters. And also I want to see as a, and let me just see, what's my next? Oh, yes. So let me go to my next slide. So I started recording animal oyster behavior this summer at Clean Valley's wet lab here in Dartmouth. And on the right-hand side, so as a professional photographer, when you're editing in a nonlinear system, you go to color grade so that you make colors more natural in case your camera system didn't record it at the beginning. So what I wanted to see was if we could actually, from the white background that the oysters were in front of, could we actually get a value for the red, green, and blue spectrums in that image, which then we could relate to algae in the shot and potentially its chlorophyll value. So anyways, I'll move on to the next slide and let Najwan pick up from there.
Okay, hi everyone. I'm Najwan, and initially when I came into the project, Amos had just completed the mussel detection system. But then because I was not very conversant with it, I was able to play around with the mussel footage, train the mussel model, and then see how that works in relation to the oysters. So initially, the mussels data set consisted of 9 mussels. As we can see on left-hand side, as compared to the oysters, which is the middle footage, there were 6 oysters. Another thing also was getting to know or understand the differences between mussels and oysters, because given my background, I just did one computer vision project, but it was not based on ocean sector, or I've not done or undertaken any kind of project in the ocean sector. So this was quite interesting for me to step foot in and then just practice and play along with. So whilst I was playing along with the mussel footage, I was realizing that there was a lot of instances where, as Lawrence mentioned earlier, that the data was skewed in a sense that there was a lot of open as compared to closed, and we just weren't understanding why that was happening. And in almost all the footage of the mussels, there was a lot of algae in the box that the mussels were being inbred in. So this raised a couple of curious questions, and Lawrence did mention that he wanted to understand why certain behaviors were apparent in mussels versus oysters. So that's what spun the mussels to oysters project. So during the oysters project, what happened was during the accumulation of data, it underwent several stages with the model. What happens is I get the data, which is recorded in video footage. And in some cases, the video footage is over an hour long, they're pretty long. And sometimes that's too big to label, especially given that when video footages are over an hour long, sometimes the objects in them don't really have that much movement. So if you're going to label that kind of data, it's going to be redundant data. So what happened is we'd skim through the hour-long footages, see what part of the video had more variations in terms of movement, and then cut that video to that long. And then afterwards, I extracted 1 frame per second from that video. So it means that for every second of the video, I got 1 still shot or 1 screenshot. So I did that for all the footages that came through. That led to the pre-processing of the data, which is the labeling. So after I got the still shots of the footage, what happens is I now want to go label if they were opened or closed, and then I then take it to go train the model. Now, this is a very important step because based on how you label it, the accuracy of how you're labeling it as a person before you put in the model, it's going to affect the accuracy or the prediction accuracy of the model. So during the training and then the retraining process, it was a redundant, the same process all over again, but this time you're just playing with different data. Another thing was when you're training a model or when you're creating a model, the most important thing is to distinguish between your training and your validated data sets. So in this instance, I spoke to Amos about how we're going to share the data, and we came to a conclusion that we're going to do 70% for training, 20% for validation purposes, and then 10% for testing. Now, what testing means is that when we finish training a model, we get undented data or data that we have not touched, labeled, or processed. We put it back into the model, and then the model then goes and then spits it out back to us, and we get the middle picture, which is the prediction accuracy from it. So from the middle picture, we can see that it's saying some of it is open at 0.75, which means that it is 75% sure that that oyster is open, or if it says closed at 0.84, it means that it is 84% sure that that oyster is closed. So that is the way that the model was training. The model was being trained on the set footages. But when we're playing around with the data set, we're getting to a point where the model was getting used to the kind of data we're feeding it with, because like in the middle picture, there were just 6 oysters. Yes, their seating position were changing, but some of them, it was just the same. And there was an instance where Lawrence and I, in a week's worth of footage, we realized that at some point, all the oysters just shut close. And we were asking each other, like, oh, like, I wonder why that could happen. And then we realized that what had happened was the sun had set, and then the reflection of the sun setting hit the glass, and then all the oysters shut close. So then that's when we realized that, okay, there is some instance where oysters are very sensitive to light and extreme exposure of light. So then that's what caused us to start going into the color grading system. We wanted to figure out from the footage and then how the algae was in the water, how were we able to tell oysters' behaviorisms and their patterns? What ways could we read that? And this was a different ball game for us to play with, because I have never played with anything that has to do with colors in terms of photography. Lawrence had more background in that, but in terms of coding and programming, it was very new to me. So we had to also invite in a color specialist who would kind of like advise us on ways to go about the project. And whenever we were doing something or whenever a coding sequence was being created, he would go through it and see if it was headed towards the right direction in order to meet the requirements we wanted it to meet. Lawrence, next, please.
Just a sec here. There we go.
Okay, so there we go.
Sorry, let me jump back.
Yes.
Away.
So in the first video, that's what the box looks like with the inbred oysters. And in the first picture, sorry. And then inside that is a color box or a color frame. Now, which is the same as shot in the second picture. If you can see, it's very saturated with algae, but if you can pick out the white disc frame from that, you'd be able to tell. Now, the reason why we used a color framing box is because a white disc has the most accurate RGB values when reflected on it. So what that means is that when you put a white box inside any kind of color box, the reflections of the color is stuck to the white, and then now we can extract values from a white background. Just like how, for instance, if you wanted to take any kind of a passport picture, it was intended that you stand behind a white frame, because it wants to pick as much accuracy of definition from the camera as much as possible. So that's why the white frame or the white box was really intended to use as our region of interest for the color grading process. Now on the third picture, you can see that that's what the colors look like, but inside the algae saturated water, it's very difficult to pick up some of the colors from it, but we can obviously tell which one was the white. So next. Now, the color grading went through several stages. Initially, when we're looking at the color grading, we wanted to pick up mathematical values of the RGB, which was the red, green, and blue values. And then using that, we're going to convert those values to CYMK, which was cyan, magenta, key, and I believe, what was the third one, Lawrence? The fourth one?
I'm going to forget offhand at the moment.
Oh, cyan, magenta, yellow, and key. That's correct.
Right. There
Yes, right. So we wanted to use the RGB values to convert to get the CYMK values. And then through that, we're going to write a coding sequence that would be able to extract those values, do the mathematical convention, store those mathematical values, and then put them in a CSV file that could later be translated and used into the actual model of the oysters. But again, like I said, it was new to me. I didn't know what to do or how to go about it. So we started by playing with the footage. That was the first thing I wanted to do. I spoke to Lawrence. He did mention that there were a couple of editing softwares where you could just put a footage in and then it would take out the values. So this was a screenshot of where we put the frame, the color box into the video, the editing software, and then we were able to pick up some of the RGB values. We could read it to some extent, see the variations, and then we could tell that, okay, using the editing software does give us some kind of idea of what to do or where to go about it, but it still doesn't give us definitive values. So what that meant was we had to create our own coding sequence, and that instead of we put the color frame or the color board frame in the video, and then we shoot the video and then we put it. So what happens is the coding sequence worked in such a way that first things first, it took the white frame. So that was our region of interest, as mentioned earlier. That was our intended region of interest. So first we bounded the white from the whole color disk, and then we loaded the video. And then when we loaded the video, we took the white frame. After taking the white frame, what we did was that the coding or the program started reading the RGB values per 1 frame per second. So if the video was, let's say, 20 minutes long, it would, for every second, it would read the average RGB values. So after the average RGB values were read from the region of interest, what happened was a coding sequence was also used to generate the CYMK. So using that, we converted RGB to CYMK mathematically, and then also we got those values. And after those values were got, and then what we did was we had the coding sequence automatically create a CSV file, generate the collection of the previously stored values in the cell, and then we were able to save that to a CSV file for future use. So during my end of tenure, that was where we were able to stop at, which was the collection of data or mathematical values of the colors. And we weren't necessarily able to, I would say, certify our assumptions just yet because the color grading was just very new. We didn't do a lot with it, but we did get to a point where we could withhold the data for future purposes in such a way that it was streamlined, easily done, where we didn't have to like read each frame individually and type it. Also, it also kind of reduced mathematical errors when recordings were being done because we're dealing with a lot of zeros and decimals. So it was very important that it was streamlined in such a way that it's easier for every footage, because mind you, this is going to be done for every footage in which the algae is saturated or not saturated. And then we're going to use that to compare the variations in footage. So in a case where maybe algae was overly saturated and this was the values we were receiving, how were the oysters behaving? Okay, this video, it wasn't so much saturated. These are the values. What was the behavioral pattern? Obviously, with time, you will start to see patterns and signs and consistencies. And that was what the color grading was looking to do. So yeah, that's it. That's all for the color grading.
That's great. So that, yes, again, so we've figured out how to at least get values for RGB. We now can look at the animal behavior, and now it's a matter of linking our system, particularly in the wild, because what's coming up in the oyster industry these days. So just because we're running over a little bit of time, I'll just quickly go through. So what we're looking to do next within IntegraSEE is we'll be working with Atlantic Aqua Farms. We're going to be looking at the larval stages. They're going to be providing us the footage of larvae as they develop, because the idea is one counts is a big deal. Just knowing how many animals you're producing in a hatchery. Secondly is the quality of the animals. Does this batch look like it's going to go through and become seed and continue through the production process, or does it have all the indicators that it's not likely to survive or have low survivorship? So we're looking at that next. The microscope on the left, being in a couple of hatcheries, their camera systems break down very fast because of the salt in the air and the humidity. So we're looking at a camera system to deal with that, plus building the models around to make all the recognition components. The second slide, or the second image, that's back in Portugal. So instead of a tank with algae in it, what we want to do is the W represents the wastewater from the fish, A is the algae medium, and J is where the oysters are growing out. So water sample from each. Again, is create a standardized shooting methodology that you can just use a smart phone to record the images, but it's standardized. You get the right or consistency between lighting and where the objects are. So we can do, just like we do with oysters and mussels, set up our boundary boxes in the same spot each time to record. The third one is because there's a disease called MSX. It's a parasite within oysters. It's been, you may have heard, it's been found in PEI now. It's been there for 22 years in Cape Breton, but this has going to have a huge impact on the oyster industry in PEI, who's the second largest growers in Canada of oysters or producers. What I want is, in the same in Portugal, whenever they have dead animals, is split them open and then take a photograph of them. And again, with a color card and grayscale, that we may be able to, in future, learn more about the parasite and its effects within the animal's tissues, just from this basic image, versus usually what they do is microscope histology and PCR analysis. The last one is on the right is actually a heart sensor that can go on the mussels, or mussels and oysters, that fits onto the shell. We've built it, but we have yet to test it, and love anybody to try this out to see what sort of data we get, if at all. The last one, which is as we develop data and want to work with the oyster growers, my slide will advance here. There we go. Is to create a 24/7 GPT chat cloud-based service, so that oyster growers who are having challenges on either putting in information to get loans, or when Hurricane Fiona came through, they could all apply for funds to compensate. Well, some of them still just under half of them still haven't received their funding back, even though Hurricane Fiona was in 2022. So they need this database. We want to develop this database, which then works with inventory in the production, finances within their facilities, but then also on the biological side, we can introduce satellite data, their own data from the farmer, as well as our biosensor. So that's our big picture in the future that we need to develop. There's somebody doing this similarly in Aqua Connect in India. By just having the satellite imagery and the farmer's data coming into a platform like this, they've increased production by 20%, reduced input costs by 30%, and been able to reduce the cost of financing through untraditional financing. So it's been a very it's been a great tool for the farmers, and they don't pay anything. They've worked out the model so that the farmers actually don't pay any money into this. It's through up and down line suppliers. Anyways, that's it for our little show for the moment. So if you have any questions, I'd be happy to answer, or Najwan, I'm sure.
I have a question.
Okay.
So I was wondering if you were focusing on saltwater, like mussels and oysters, I'm assuming, are you able to apply this with freshwater species as well?
Yes, and in one of those pictures, there was I threw it at a little sideball to Najwan. So I said I went out and collected a freshwater mussel from lake around us here, and I put it in a tank and then just recorded and said, "Oh, here's some footage. See if your model can work with a freshwater mussel." And sure enough, it did. It worked just fine. So we would like
Amazing.
to. And I didn't mention too, we were sent a kit. So instead of Hall sensors at North Carolina State University, there's a researcher and some graduates who are using accelerometers instead. So they find it's easier math to determine the degree of the shells opening and closing. And that was all based on looking at why mussels are disappearing in our lakes, which is happening big in Europe and in North America. So yeah, we'll do both. We're just focusing on what's available to us now and the easiest to answer.
Okay, amazing. And also, how did you decide on the probability threshold? Like, how did you decide when the model was what you considered accurate enough at determining whether the mussels or oysters were open or closed?
So what happens is when you have to feed the model after it's been trained with additional footage, right? And then it's going to tell it the way the model has been trained is it'll show you like the bonded frame, it writes it. And then you have to use your human eye to assimilate if that is correct or not. So for instance, if a mussel or an oyster was visibly closed and then the model said, or it read it as opened at 84%, you can tell that there is like a serious problem going on with the model. But sometimes you can tell, and then depending on also how light or bright the footage is, we were also intended to train the model in such a way that I could also tell from that instance, but you also look at the prediction loss and prediction error as well from the module. So doing all of that, we could tell that, okay, the model was also going into a really great place. And then it peaked at, I believe, 96%. So giving all the data we had, and then we just decided that the model was doing great as it was right now. And then we switched to color grading.
Okay, great. Thank you.
No problem.
Are there any other questions from participants?
And I'd just like to say, if anyone has other questions later on, do feel free to contact me or to learn more about the marine environment and the applications of AI. I'm happy to chat with anybody.
Yeah.
Amazing. Okay, well, if there's no more questions, thank you for taking the time out of your busy day, Lawrence and Najwan. We really appreciate
Thank
it. That was
you.
a fascinating talk. So I appreciate it
Thank
a
you.
lot. Lawrence and Najwan, do you have any last words before we wrap up?
No, not really. Just that this was a really fun project to work on and it was great.
Amazing.
And I'd just like to say it's taken the visions that I have, partly as a storyteller and where to move through sort of scientifically and gone, it's really been fun. Especially, I would say the biggest one was the color, as a, can we turn what, I was even going to take
Sure.
just pictures off our editing suite and say, just take these pictures and analyze them. But to actually get solid data out of that's a big deal.
Yeah.
We tend to work more in the qualitative area when you're working in behavioral analysis. So hard data is really nice to be able to generate. Plus, we've also then got temperature and salinity sensors on board with the system.
Mm-hmm.
So we can now also provide more environmental data to understand what's going on.
Wow, that's incredible.
Yeah.
Well, thank you again to everyone who joined and to our presenters, and I hope you all enjoy the rest of the day, and hopefully we see you at the next discovery session.
Thank you. Bye.
Bye.
Bye.